Assalamu alaikum. We will be starting in a new chapter, Chapter 2, DNA, Genetic Information and the Cell Cycle. For the first lesson, Activity 1, the karyotype. At the end of this video, you will be able to describe how to obtain a karyotype and to define the karyotype. Make sure to prepare the extra worksheet and a pencil case. Let's return briefly to the title of this chapter, DNA, Genetic Information. And if we wanted to study the DNA and genetic information, we need to find where they are. Well, you've learned previously that this DNA is found inside the nucleus in molecules called the chromosomes. So how can we study the chromosomes of an individual? The answer lies in the karyotyping technique. Since karyotyping is a technique, there must be a procedure to perform it. This document shows the procedure for performing the karyotyping technique. As this document shows, a blood sample is collected from an individual. The cells collected from the blood are then cultured, and colchicine was added to those cells. After that, the cells were placed in a hypotonic solution, and then the cells were split on a slide, treated with chemicals, and observed under the microscope. Then, a microphotography of the chromosomes was obtained, and then those chromosomes were karyotyped. This is simply how to describe the procedure of karyotyping technique. Now let's see if you followed correctly each step. Try to find the missing step in this table. If the first step is to collect blood, what is the second and the third step? As we said, the second step is to culture the cells and then add colchicine. Then, put the cells in a hypotonic solution, spread the cells on a slide, treat the chromosomes with chemicals and colorants, observe the chromosomes, then take a photo of them. Finally, cut the photo of chromosomes, then arrange the chromosomes in pairs of homologous chromosomes, or karyotype them. These are, in short, the steps of karyotyping technique. Now, what is the importance of each step? What is the importance of colchicine? What do we mean by culturing the cells? Now let's find the importance of each step in karyotyping technique. As we said previously, first a blood sample is taken from an individual and then cultured in a growth medium. This growth medium will trigger the cell to start mitosis or cell division. Then a chemical substance called colchicine is added. The importance of this colchicine is to stop mitosis at metaphase. Now watch out carefully what will happen next. A salt solution or hypotonic solution is added to those cells. As you've learned in the previous lesson, hypotonic solution will lyse the cell, which will get rid of the plasma membrane. Those cells are stopped at metaphase. Let's pause for a moment and think here. If those cells are at metaphase, then the chromosomes are far from each other. Each one is separate from the other. Now for our final steps, those cells are spread on a slide and certain chemicals and colorants are added. The colorants allows the banding of chromosomes, as you can see in the image. Then this slide is observed under the microscope and photographed in order to get an image of chromosomes. The image is then cut to separate each chromosome and to get a final karyotype. Did you find the importance of each step in karyotyping technique? The idea is very simple. Blood is collected in order to get nucleated cells, and of course those cells are white blood cells. Since the red blood cells are anucleated, they do not have a nucleus. The cells are cultured in order to increase the number of nucleated cells by mitosis or cellular division. Colchicine is a chemical substance that is added to stop the division at metaphase. At metaphase because at this step chromosomes are clear and separated in order to be seen. And as you've learned in the previous lesson, a hypotonic solution allows the water to enter the cell and the cell will explode, which exposes the chromosomes for us to see them. The cells are treated with colorants in order to obtain bands. And cutting the photo of chromosomes and separating and arranging them is another term for karyotyping. Now here's the new question. If you're seeing those chromosomes under the microscope separated in this form, how are they being karyotyped? How are they being arranged in a karyotype? Let's start by observing the following karyotypes in order to find the criteria used to prepare a karyotype. The first obvious thing you're seeing that chromosome number 1 is larger than chromosome number 21. So we can say that the first criteria is the size of chromosomes. And how are they arranged according to size? They are arranged in decreasing order of size, where chromosome number 1 is the largest and the size decreases until we reach chromosome number 22. For our second criteria, let's observe clearly chromosome number 9. You can see here we have like black spots. Those two black dark spots, we call them centromeres. So for the second criteria, chromosomes are arranged according to the position of the centromere. And you can see here that each pair of chromosome has same location of the centromere. For our third criteria for preparing a karyotype, let's observe chromosome number 12. As you can see here, chromosome number 12 is colored with bands, where you have the blue band, the red band, and the green band. And those two chromosomes, they have the same banding pattern. So thirdly, let's say chromosomes are arranged according to the banding pattern, where those two chromosomes at the same number have the same banding pattern. Those chromosomal pair having same size, position of centromere, and banding pattern are called homologous chromosomes. Homologous chromosomes are pair of chromosomes having same size, centromere position, banding pattern, and gene map. Now, gene map, we mean by it genetic information. To sum up what we've learned, in this karyotype, chromosomes are being arranged according to the size, the position of the centromere, and the banding pattern, where each pair of chromosomes represent homologous chromosomes. However, we have some kind of contradiction here. You can see that all these chromosomes here represent homologous pairs of chromosomes. However, for these two last ones, the Y and the X chromosomes, they are non-homologous. The shape of the Y chromosome differs greatly from that of the X chromosome. Here we have a new rule to add. All chromosomal pairs from 1 to 22 represent homologous chromosomes called autosomes. However, the last two chromosomes, which are called the sex chromosomes or the gonosomes, are of two types. They can either be homologous XX or non-homologous XY. Why are they different? Because those gonosomes gives the gender of an individual. For XY, the gender of the individual will be male. For XX, the gender of the individual will be female. Let's sum up all the information we've learned in this lesson in order to define a karyotype. A karyotype is a photograph or a drawing that shows the set of chromosomes of two types, either autosomal or gonosomal chromosomes, of a living thing where homologous autosomes are arranged in decreasing order of size. We must stress here on the term homologous autosomes because the gonosomes can be either homologous or non-homologous. In this video, you've learned to define a karyotype as a photograph for different autosomal and gonosomal chromosomes arranged according to different criteria, and that this karyotyping technique is a set of steps, each step with a certain importance. As an assignment, please make sure to study the summary sheet and to solve and correct Part A of Worksheet 4. This video is prepared and recorded by teacher Zahra Shari and supervised by teacher Ehsan Hassan. Thank you for your time.